There are a variety of different mutations that can occur in DNA or RNA, mutations in the nucleotide sequence. The main thing that I guess I'm thinking of are mutations in the germline. So as an adult, you can have mutations in random cells in your body, in either DNA or RNA, but only mutations in the gametes or cells that produce gametes will actually be a heritable mutation. So here's mutagenesis. It's just the same thing I just said. Genetic information changing from mutations. So originally, your life, I guess you might say, is building nucleotide sequences from scratch. But at this point in evolution, in the history of Earth, it's all modifications of existing sequences, duplication of information plus slight modification through mutations. So one thing that life does during meiosis, that you get different chromosomes from each parent in a diploid organism, such as a human, and you're able to, in that process, the chromosomes swap sections to kind of shuffle the genes around a little bit more. So I'm pretty sure that's still considered a mutation, even though it's kind of an intentional through evolution process of just swapping large sections of chromosomes around. I think what most people think of as a mutation are point mutations. They're just a single nucleotide changes to something else, a single nucleotide polymorphism, if you will. So if you're focused in on a certain type of mutation, you can look into a mutation rate, just how often that mutation occurs, and that might be somewhat of a function of the environment. If you're out in the sun, you might get more mutations on your skin cells, but those wouldn't be heritable mutations. So I guess just looking at the germ cells and the heritable mutations, there's also going to be certain rates that are vary over the genome. So different parts of each chromosome will have different rates of mutation. So the human mutation rate is about 64 new mutations per generation. So just on average, you can expect that you and your parents have, you got DNA from your parents. It's all shuffled around through crossing over of chromosome sections. But then just looking at probably the single point mutations, you're looking at about 64 changes from your parents' DNA. I guess you could probably say, your siblings are going to have different shuffled genes from your parents. So ignoring the shuffling, you probably have 64. It'd probably be 128 changes between you and a sibling, ignoring the shuffling of the chromosomes. Then, like I just mentioned, the germ cells are the sperm and egg cells. They probably give rise to sperm and egg, the gametes. So that's where you're, looking back at evolution and trying to understand how organisms and species change over time. You're looking at only changes in germline. And then each change will be occurring without knowing what it's creating. So there's a mutation rate, but then there's various types of mutations, like silent mutations, which kind of go unnoticed in the phenotype. And then you have genes that create proteins, eventually genes that don't create proteins. So any kind of mutation can have a variety of effects, negative or positive or neutral. So you might have a sequence of nucleotides that are conserved. So you have some mutation rate, but it doesn't mean that you're going to see that same mutation rate on each gene, because certain genes can't handle mutations. They are devastating to the organism, or just slightly less, slight changes in fitness to the organism can show up over large stretches of time. But then there's certain sections of DNA that can kind of just take any mutations without consequences to the phenotype. And then there's mutations that are induced by, say, an experimental scientist. So people in experiments can use chemicals or UV light or any kind of either even genetic engineering to just change the sequence, mutate it, or break the DNA strand and insert some other sequence in the break. That's a little bit different than the just mutation side of it, but that is also a way that DNA sequences can be altered. And I'm assuming there's very similar methods to change RNA if you wanted to do that as well.